Hello, everybody, and welcome to today's webinar, Using Artificial Intelligence to Disentangle Glaucoma Pathogenesis with Dr. Luis Pasquale. My name is Elena Sturman. I'm the President and CEO of the Glaucoma Foundation. Since our founding in 1984, the Glaucoma Foundation has worked to encourage and support basic and applied research in glaucoma with the goal of preserving and restoring vision. Our board of directors and scientific advisory boards are dedicated to identifying the most promising avenues for research. Our annual think tank brings together scientists from around the globe and from different fields to address the problems of glaucoma. The Glaucoma Foundation has given millions of dollars in seed money for innovative projects, and we continue to provide a space for scientists to think creatively and gather the data they need to pursue major grants from entities such as NIH and NEI. Above all, TGF is dedicated to improving the lives of people with glaucoma. We strive to be a valuable resource for education and support of our patients and caregivers. You will find a wealth of information on our website about glaucoma, its diagnosis and treatments, and the research that we support. Today's webinar is sponsored by Santen. We are so grateful to Santen for this sponsorship and for our incredible partnership. We also want to thank Dr. Pasquale for making this presentation today. Dr. Pasquale is Shelley and Stephen Einhorn Distinguished Professor of Ophthalmology and Site Chair of the Department of Ophthalmology at Mount Sinai Hospital. He is Vice Chair of Translational Ophthalmology Research at Mount Sinai Healthcare System. Dr. Pasquale is a member of Journal of Glaucoma, Ophthalmology Glaucoma, Asia Pacific Journal of Ophthalmology, and American Journal of Ophthalmology editorial boards. He currently serves as glaucoma trustee for ARVO and is a member of the National Institute of Health National Advisory Eye Council. Dr. Pasquale is NIH Principal Investigator with a continuous support since 2006. His research focuses on the discovery and primary prevention strategies in open-angle glaucomas, and he has been a co-investigator on many research projects to assess the use of artificial intelligence and machine learning algorithms in glaucoma. Dr. Pasquale is a member of the board of directors of the Glaucoma Foundation and is a co-chair of the Scientific Advisory Board. Before we begin, I'd like to remind everybody that if you have a question during the webinar, you can submit it by using the chat button on your screen. We will do our best to answer it during our Q&A at the end of the presentation. And now it is my great pleasure to welcome Dr. Luis Pasquale.
Thank you, Elena. Thank you for that kind introduction. And thank you everyone for joining this webinar this afternoon. I'm excited to speak with you all today. And just trying to get my slides up here going and going to full screen. I understand that this audience is large, and is weighted more toward patients than researchers. But I try to make this talk have something for both groups, but keeping in mind that there's a largely audience here. So let me get started. And I'm excited to talk about this. And let me first mention my conflicts of interest and grant support. As Elena said, I am supported by the Glaucoma Foundation, NEI, National Eye Institute, Research to Prevent Blindness. And I'm also consultant to various companies. I'm really fortunate to have the research support that allows me to explore issues related to the pathogenesis of primary open-angle glaucoma. Now, many of you know that glaucoma is a disease of the optic nerve, and that primary open-angle glaucoma is probably the most common glaucoma subtype in the world. Right now, it's estimated in 2021 that there are about 53 million cases. And it's fair to say that strategies to prevent glaucoma from developing don't exist at this point. and that this talk is going to review how we're using artificial intelligence, not necessarily to diagnose glaucoma, but to understand what's causing it in the first place, which is a fairly novel use of artificial intelligence. As I said, the optic nerve is a telephone cable that connects the eye to the brain, and glaucoma is a disease of that telephone cable. And this slide gives you a schematic of how that telephone cable connects the eye to the brain. And you notice that the connections are quite complex. The researchers in the room know about this. But the point is even if those of us who do research are trying to rejuvenate the optic nerve, to get nerve fibers to grow back to the brain, it's a daunting task. There's a lot of complex connections here. And even if you're able to accomplish this, you're still going to need to understand what caused the optic nerve to deteriorate in the first place in order for those strategies to be successful. Now, just to go a little bit further about this telephone cable has a telephone number, and that the optic nerve is more than what the ophthalmologist sees here when they look into the back of your eye. This is an optic nerve of a patient with glaucoma. What they're looking at is this portion of the optic nerve, but the optic nerve is about 55 millimeters in length. But we know that the glaucomatous process seems to affect the optic nerve right about here. This is what we call the intrascleral portion of the optic nerve. Now, once I was giving a lecture to residents trying to explain what happens in glaucoma, and I was saying that in glaucoma, when the optic nerve deteriorates, you get excavation of the neural retinal rim. And what I mean by that is that if you were a little man sitting on the surface of the retina, you could stick your thumb underneath this space, which we call the neural retinal rim. This is what we mean by excavation. And one of the ophthalmologists in the audience was a cartoonist wannabe, and he made this cartoon of what I was describing. So I'm grateful to Dr. Mark Kupawasser for generating this cartoon. And what we mean is really translated here. So this is an actual appearance of a glaucomatous optic nerve. And if you look at this retinal vessel here, it looks like it disappears and then reemerges onto the surface of the retina. That's because the rim here has been excavated, undermined. That vessel is going underneath and then reemerging onto the surface of the retina. And to this day, we don't understand exactly why this undermining occurs. Of course, we blame it on pressure inside the eye being high, but it's probably way more complicated than that. So let me present a case to you to illustrate why we need glaucoma prevention strategies. So this is a patient who presented to me fairly recently. He's 39 years old. He said that he noticed some blurring of his vision near his nose. And lo and behold, his intraocular pressures were very high. And as you can see, these are the visual fields of his right and left eye. And they show, unfortunately, an advanced amount of glaucoma. And we were able to lower his intraocular pressure into a reasonable range. However, this field loss is going to stay with this patient for the rest of his life, and it's certainly visually disabling at this point. He can no longer drive, which is what he was doing for a living. And the issues here is, while we can't bring back the vision that he's lost, how did this happen to him in the first place? And could we devise strategies to prevent this from happening to him in the first place? And there is a lot of patients like this. This isn't just an isolated story. Very frequently, we hear that glaucoma is the thief of sight. You don't know you have it until it's quite advanced. And so we really would benefit from having any strategies that would reduce the rate at which this happens to patients. And so this is what my research is about, to try to find those kinds of strategies. So how do we use artificial intelligence to help us understand what's causing glaucoma in the first place? Well, let me first start by saying, give you a typical example of how artificial intelligence is being used right now in the field of glaucoma. Right now, what we're doing are things like this. We take a whole large sample of pictures of the optic nerves, and we label them as having glaucoma or not. We develop these computer algorithms to learn which of these images have glaucoma and which do not. And after a certain period of time, these algorithms can be better than the actual human examiners at saying whether or not a typical photograph of an optic nerve is glaucomatous or not. And that's what this is demonstrating, is how these computer algorithms go through a process by which it can determine whether or not an optic nerve has glaucoma or not. And this is really amazing that we can train a computer to be better than us in terms of detecting glaucomatous optic neuropathy. But I will in due course show you how we're using artificial intelligence. Now, in order to understand the pathogenesis of primary open-angle glaucoma, which is a disease where the pressure may or may not be high, but yet this sort of glaucomatous deterioration occurs and ultimately leads to visual field loss. How do we sort of schematize this disease? And this is important. So let's assume for a second you're born with a normal optic nerve, but you're destined to develop primary open-angle glaucoma. There's a preclinical phase of the disease where there are biological changes that occur. We sense that this preclinical phase of the disease is prolonged, insidious, has no symptoms. And after a while, though, it triggers the clinical onset of the disease. There might be early visual field loss. There might be subtle optic nerve cupping that occurs. And then as the disease progresses, the patient becomes symptomatic, and then it ultimately leads to blindness. And the importance of this slide is twofold. First is to appreciate the fact that the natural history of primary open-angle glaucoma is not something that plays out over days or weeks or even years. It plays out over decades. We have some sort of cartoonized versions of a time course for this disease and this patient occurring from 1990 and developing advanced loss about 30 years later. That's one thing. The other thing is we don't know what's going on during the preclinical phase. We suspect that there are biochemical changes that are occurring that trigger the glaucomatous process. We have no idea what those things are. And that's what we think we need to focus on if we're going to develop disease prevention strategies, right? If you want to find out how to prevent the disease, you need to find out what's happening during the earliest phases of the disease. And that's what I've been focused on for my whole career. And luckily, I have access to an incredible patient resource up at Harvard, where we follow very large cohort of health professionals, both nurses and male health professionals. The sum total is 270,000 people, and we've been following them since 1980. So we've been in the game for the long haul here to understand the natural history of glaucoma, particularly during the early phases of the disease. And this large cohort of patients have dutifully filled out every 2 years surveys about what they eat, what they drank, where they live, did they exercise or not, what diseases that they were developing, including glaucoma. And so we had that data available to us. Luckily, about 1/3 of these patients donated blood to us back in the period between 1989 and 1993. When that blood was collected, we basically processed it immediately, extracted the DNA, and extracted the plasma, froze it at minus 130 degrees Fahrenheit, put it in a freezer farm for waiting for the day for when technology would catch up and allow us to leverage that piece of data to help us understand what's going on in primary open-angle glaucoma. And that day is here, which is exciting. And so we have contributed the DNA from those patients in a global effort to resolve the genetic architecture for primary open-angle glaucoma. This is something that started in 2010 when the ability to genotype patients at a very high number of genetic locations throughout the genome became available. But this is a global effort. Not only are the health professionals in there, but there are patients in there from all over the world. And so this is part of the International Glaucoma Genetics Consortium that's led by David Mackey from Australia and Janie Wiggs from Harvard. And it's important to know how genetics relate to primary open-angle glaucoma because you're born with your genes. And so your genes are going to give you some insights into what's happening during the preclinical phase of the disease, which is what we're focused on, right? And in 2021, we've published our latest results on finding genes for primary open-angle glaucoma. And there are over 120 genetic loci that are associated with the disease. Now, this figure here might seem a little disarming. Don't be intimidated by it. Basically, the x-axis lists the chromosomes, starting from the first chromosome right down to chromosome 22, and then the sex chromosome. You have 23 chromosomes. And then on the y-axis, we're depicting locations in that genome that have loci associated with primary open-angle glaucoma. So locations that rise above this horizontal red line here, those are locations where there are genes associated with primary open-angle glaucoma. And you can see that there are a lot of them. And it's hard to figure out what all of this means. This is what we call a Manhattan plot. And it's appropriate because the Manhattan plot of where the genes are located for primary open-angle glaucoma looks just like the skyline of New York City. Complicated and crowded and a lot of players. And the name of the game is to try to understand how these genes are contributing to the pathogenesis of primary open-angle glaucoma. Now, these 127 genetic loci, by the way, that list is expanding as I speak. We're working on another project now where that list is going to above 300. But they only explain 9% of the primary open-angle glaucoma heritability. And it is difficult at this time to put these genes into a framework so that they explain the pathogenesis of the disease. So this is where I want to introduce the concept of artificial intelligence. So to begin to disentangle what's going on, what we've decided to do is to use artificial intelligence as a tool to objectively determine different patterns of visual field loss which develop in early onset primary open-angle glaucoma. And many of you who have glaucoma, you know what a visual field is, you've done a visual field, you may even be somewhat familiar with what the visual field printout looks like. And this is a typical visual field from a normal patient, which has normal reliability parameters, and it gives you some assessment globally as to whether or not the visual field is normal or not. This GHT stands for glaucoma hemifield test, being within normal limits, but it tells you nothing about if the field is abnormal. What is the regional nature of the disease? Is it located more in the superior peripheral field, the inferior peripheral field? Is it located in the central visual field? It doesn't give you any of that information. And so Tobias Els from Harvard is a bioinformaticist who developed an artificial intelligence algorithm called Archetype Analysis. He didn't develop this, but he applied it to visual fields to decompose them objectively into their different components so that they could be quantified and analyzed. And I thought this was great because primary open-angle glaucoma is such a complex disease. I showed you the Manhattan plot where there's so many genes that are related that this disease is probably not one disease, but multiple diseases. And here was a nice tool that allowed us to break primary open-angle glaucoma down into different functional parts. And so just to give you an example of how archetype analysis works, this is a visual field from a patient with glaucoma who does have functional loss. And basically what archetype analysis does is it takes a visual field and it breaks it down into a linear equation. So what this is saying is that this visual field is 23% of this normal archetype, that's this one right here, and that is 34% of this archetype, archetype 14, where there's early paracentral loss that breaks out to the nasal periphery, and it is 43% of this superior nasal loss. So this is great because the computer is doing the work for us. It's doing it objectively, and we have validated that this is a robust algorithm. We've used these coefficients to follow patients and prove that these weighting coefficients can tell us about visual field progression. So what we did was we have in our health professional cohort over time, we've collected nearly 2,000 cases of new onset primary open angle glaucoma from health professionals throughout the whole United States. We took all of the visual fields from these patients from the earliest date that we could find that when they developed glaucoma, and we applied this archetype analysis to those visual fields. Say, what does primary open angle look like in the functional domain at its earliest onset of disease? And this is what we found. Archetype analysis discovered 14 different patterns of visual field loss. Red's bad, white and blue is good. So, you know, there are some people that have little to no visual field loss. There are different patterns here showing different patterns of loss. This is not surprising given the complexity of primary open angle glaucoma. But I want to point out two additional things. One is that, and most importantly, is that this archetype analysis affirmed that this early onset paracentral loss occurred even in new onset glaucoma. This is important because we saw this pattern in glaucoma clinics, but we weren't sure that we would pick this up in people who were newly diagnosed with glaucoma. The dogma in glaucoma is what I learned when I was a resident, that glaucoma is a disease that affects your peripheral vision, and with time, that peripheral vision sort of migrated into the center, and that's how blindness occurred. But this is saying that there is a significant subtype of patients whose damage starts out in the center of their vision. That's number one. Number two is that, remember that these visual fields all come from health professionals who have good access to the healthcare system. Yet, even amongst these patients, several of them are presenting with advanced patterns of loss, just like that patient that I presented at the outset of this talk, that 39-year-old patient who noticed symptomatic glaucoma, right? He was at the end stage of his disease when he was first diagnosed. So we see that too, even among health professionals. Now, the pattern that I'm focusing in on is this paracentral loss as demonstrated here. Why? Because these patients, they tend to have eye pressures that are closer to the normal range, number one. Number two is they have early visual disability. They have difficulty reading, they have difficulty with depth perception. So their glaucoma is bothering them pretty early on. And number three is we find that these patients tend to be the most rapid progressors. We know that glaucoma is a disease that generally is slowly progressive, but on average, 10% of primary open-angle glaucoma patients exhibit rapid progression. And these patients, it's not, hard and fast, but these are the ones that tend to rapidly progress. So we are really interested in that subtype of primary open-angle glaucoma because we think it could teach us about the general fundamental mechanisms of what's going on in the disease. And I'll show you some data to illustrate that that is probably true. So let's switch gears for a second and talk about metabolomics. So what's that? So metabolomics is the systemic study of unique chemical profiles associated with the disease. Basically, it's what's floating around in your blood that might be associated with the disease that you have. So we know that genetics is important, but metabolites are influenced both by genes and by environment, and metabolites are closer to the tissues that are involved in the disease process. So our hypothesis is that there is a chemical profile signature that's associated with primary open-angle glaucoma that could be detected even before the disease manifests itself. So that's the hypothesis that we have explored here. And so and the data I'm presenting to you right now hasn't been shown anywhere. We will be presenting it at an upcoming international ophthalmology research meeting in May in Colorado. And this work is currently been submitted for peer review. So what we did was we had found 599 cases of primary open-angle glaucoma, and the blood that we used was from before they were diagnosed. So they were not treated yet. They were not recognized as cases. And we matched them to 599 people who we knew didn't have glaucoma. They said that they went to their eye doctor and their eye doctor told them they did not have glaucoma. And so the plasma, so we used that plasma. Remember that I told you that blood was donated from 1989 to 1993. That's the blood that we used for this analysis. And so in most of the cases, the blood from the primary open-angle glaucoma patients was collected 10 years before they were diagnosed. And we used the latest version of liquid chromatography, mass spectroscopy that's available that allows us to look at large numbers of metabolites at the same time in a very small sample of fluid. And this is work that was done under the direction of Clary Klisch at MIT in Cambridge, Massachusetts. Ultimately, we were able to be confident about 367 metabolites in 17 different classes. And so we looked at that in relationship to early onset glaucoma. And this is what we have found overall. And this is a complex slide, so just let me orient you. These are the different classes, the 17 different classes of metabolites. And red means that they're adversely or positively associated with the disease. Blue means they're inversely associated with the disease. And these stars indicate the statistical significance. Now, I want to point you to this number 5. So model number 5 means we controlled, not only are the cases matched to controls very carefully, but we additionally adjusted for every additional risk factor that we can think of. And what we find is that there are some lipids that are positively associated with the disease, and that there are some organic acids like pyruvate and things like that, ketone bodies that are good for mitochondrial function, that are inversely associated with the disease. And what's really amazing is that if we limit the analysis to the cases with paracentral loss, so we drop our case number down from 599 to 178, look what happens. The statistical significance goes up dramatically. So these paracentral cases are really telling us something about what is happening early on in primary open-angle glaucoma. The relationships, the adverse or positive associations with glaucoma seem to become more striking and statistically significant for triglycerides, diglycerides, certain phospholipids that are resident to biological membranes. And the relationship, again, with the organic acids becomes very strong and in the protective direction. So this is really interesting. Now, we took it one step further. We validated this by replicating it in an external data set. So there is something called the UK Biobank, which is 500,000 people from the UK that gave blood and answered questions about lifestyle, just the way the nurses and the health professionals did. And they said whether or not they had glaucoma. They used this blood to do genetics and metabolomics on them. And basically what this slide is saying is we found over 2,000 self-reports of glaucoma with 44,000 controls. And again, we're seeing those triglycerides being adversely associated with glaucoma, while we're seeing organic acids such as ketone bodies being inversely associated. So we are basically replicating these results in another data set. So we feel pretty confident that there is an adverse relationship between selected lipids and glaucoma, and a protective effect for ketone bodies being associated inversely or protective for glaucoma. So in conclusion, elevated serum triglycerides and lower serum organic acids increase the risk of glaucoma. And that's sort of what we're thinking. And we were emboldened by the fact that we had that paracentral subgroup to help us create what we think is a new pathogenetic tree for glaucoma, that somehow elevated serum triglycerides via genetic changes and environmental changes can trigger both elevated intraocular pressure and vulnerability to optic nerve damage. So things like triglycerides, they don't just float around in your blood by themselves. They're carried on lipoproteins. And we weren't able to pick up the lipoproteins in the nurses and health professionals because the assays that we used didn't allow us to look at lipoproteins like LDL and HDL. But we were able to do that in the UK Biobank. These lipoproteins carry triglycerides in their core, and the smaller lipoproteins can gain access to the anterior chamber. And we hypothesized that some of these lipoproteins may contribute to trabecular meshwork dysfunction that leads to elevated intraocular pressure. At the same time, elevated triglycerides might reduce the levels of things like pyruvate and other ketone bodies, which are critical for the optic nerve. What this slide shows here, this is the optic nerve in cross-section, and this is a stain for mitochondria. And there's a lot of mitochondria in the optic nerve head. And remember at the outset, I said that the optic nerve head is where the damage from primary open-angle glaucoma occurs. So if there are reduced ketone bodies, there's reduced mitochondrial function, and that increases optic nerve vulnerability and could lead to damage even if the intraocular pressure is in the normal range. And so while there's much work to do here, and this is somewhat speculative, for the first time, I feel that I can say this is a roadmap to follow that will lead to more insights into the disease process and may lead to new drug targets for the disease. And certainly there's some studies now going on at Columbia where they're trying to leverage mitochondrial function to improve the outcomes for glaucoma. And those studies are exciting and interesting. And this work is consistent with that line of research. So the approach of trying to re-energize those little batteries in our optic nerve head, the mitochondria, that seems to be a viable approach to try to help our patients moving forward. So in conclusion, and I think I'm doing good on time here, which is perfect, artificial intelligence helped us disentangle glaucoma pathogenesis by affirming the presence of early paracentral loss in primary open-angle glaucoma. We found using a metabolite approach that metabolites associated with glaucoma, they were more strongly associated with the paracentral loss pattern in the disease. And altered lipid metabolism may be a key early feature in primary open-angle glaucoma. I want to thank you for your attention, and I'll be happy to take any questions. Maybe I'll stop sharing my screen.
Yes, stop sharing your screen, please. Thank you, Lou, so much for this wonderful presentation and for looking to the future. It's very, very exciting. I have one question in the chat right now and one question that was previously asked through the website. I will start with that question. What clinical trials in New York City area can we participate in regarding glaucoma and artificial intelligence?
So, Elena, that's a great question, whoever asked it. You know, one of the holy grails in artificial intelligence is to come up with a randomized clinical trial that proves that artificial intelligence improves patient care. This is something that Dr. Eric Topol, who is a cardiologist, geneticist, who's been like one of the top thought leaders in this field, has been talking about this for quite a long time, has suggested he would like to see. And right now, there isn't a single such trial in any field of medicine that has used artificial intelligence. However, I'd like to put in a plug for the Glaucoma Foundation because they have had the insight to try to position us to do such a trial. So the Glaucoma Foundation, as you well know, put out a request for funding to come up with a glaucoma forecasting algorithm. And a group from UCSD won the award. And what they're going to try to do is to come up with an algorithm that predicts which patients are going to need surgery. Now, if that algorithm proves to be true and it works, that could be the grounds for a randomized clinical trial right there. So you would apply the algorithm to patients. You would take the patients and say, okay, these are the group of patients who look like they might need surgery down the road. We'll randomize half the surgery and half the usual care, and we'll see what happens to those patients. That is how artificial intelligence could help the field. And it's going to take some time, but I think the Glaucoma Foundation has helped to position our field in order to develop such a randomized clinical trial. But right now, there isn't any that I know of.
Well, thank you so much. And of course, we will be looking forward to all the results from the research we are sponsoring and hoping that that will bring clinical trials in artificial intelligence.
Thanks.
There are lots of questions. So do you want to scan and—
Yeah, so let me start. So there's a question from Padu from Indiana University. Cholesterol esters are different. We're looking at cholesterol esters in your slide. How is the cholesterol levels? So yeah, so this is complicated. So this is sort of a question that is more of a researcher question. So you have to know the anatomy of a lipoprotein particle. So the lipoprotein particles in their core, they have the insoluble triglycerides and diglycerides. But on the outside, they have phospholipids. And in the phospholipid membranes, they have cholesterol esters that are in those membranes. And that's very different than cholesterol itself. And we have seen an inverse relationship with the cholesterol esters, meaning that they're protective, but cholesterol itself is adversely associated. Cholesterol esters might make those membranes more fluid-like. They might make them more friendly to the cells that they interact with, but we're at the beginning to really understand what's going on there. Overall, cholesterol esters right now look like they're good. Cholesterol itself might not be as good for glaucoma, but it's very complicated because of the packaging that they have in the lipoproteins. And we know lipoproteins scan, that they have sizes ranging from like 8 nanometers up to 1,000 nanometers. And so some of them can get into the eye, others cannot. So it's a very complicated process. We are applying for NIH funding to repeat a metabolomic scan on our very samples using the Nightingale platform, which will allow us to look at the lipoproteins directly. At ARVO, we're going to show the Nightingale scans with lipoproteins where they have OCT, they have IOP, and there's definitely interesting stuff going on. So I'll stop there and just say, Padu, it's complicated.
Got it. There is a
but I hope I began to answer your question.
There is a great question from one of the, I know he's a patient and a patient advocate, and I'm going to read it in its entirety. So you are on the verge of developing a blood test that can determine the risk of someone possibly getting open angle glaucoma. While that is amazing, my question is, what do you do then? Is there a way to prevent disease from happening?
Absolutely. You know, and by the way, I don't I don't intend to patent anything like that. I believe that the patients have contributed their blood, the NIH has supported these projects, and this should all be ultimately open source. I don't know if that's how it's going to pan out, but that's the goal, right? I mean, if I could draw your blood and say when you're a baby that you're going to get glaucoma, first of all, we're going to watch you a little more carefully. Number two is that it's clear that there's a lot of different ways to get primary open angle glaucoma. There's no one way. And even though I showed that last slide about an overview of what could be going on, it's way more complex. It's ultimately going to be way more complex than that. But it could lead to precision medicine solutions. You know, what's good for one person may not be good for another. And they might be getting glaucoma from a totally different mechanism. So, we do know that even if, let's say, you had a lot of genes, let's say you had 127 genes for primary open angle glaucoma, guess what? That still doesn't mean you're going to get the disease. And we're doing research about that as well. So we're funded from NEI to go into biobanks at Mount Sinai and up at Harvard and call out the people with the highest and lowest genetic risk score and see, who are these people? What do they have? And we're going to find that some people that have like a full house match of genes, they don't get glaucoma. And we want to find out why, because we don't know. And then there's going to be other people with very little and they got bad glaucoma. We want to find out what's going on with them as well. So a lot to do there.
Thank you. Thank you very much. There is another question about the percentage in your study that you used. What percentage of Asians and African Americans, if you know, out of the sample?
Yeah, so that's a good question. So in the nurses and health professionals, it's only 1 or 2%. In the UK Biobank, it's higher. And that's something that, we need to address. UK Biobank, I think it's something like, 10% are people of color. And so we need to do more of these studies in people of color. Mike Hauser is leading an effort of Eyes of Africa. And he's been very dedicated to glaucoma in African-derived people. And I got to get back to talking to him about it. I promised him I'd help him out with the project. But we need to do more of that. We need to do more of this same exact study in people of African ancestry. The reason why so few people of African descent are in our study is because the people entered the study were health professionals in 1980. And we weren't thinking a lot about health equity in 1980 and trying to encourage people of color to go into the health professionals. We do a lot of that now. But in that time, it reflected the people who entered the professions at that time. So that's why we only have 2% African-derived people. But guess what? Even with that 2%, we see, for instance, that African heritage is a risk factor for glaucoma. Using that archetype analysis, we see that people, African American health professionals, were the people that were presenting with advanced patterns of loss. So we do see things, but on the genetic and biomarker side, there's still much work to do. We need to increase the samples in order to address those questions.
Thank you, thank you. Here's another good one. Would you suggest that the general population, when seeing their primary physician for their annual physical, that they add a screener for open-angle glaucoma?
Yes, they should, and it's going to be fairly easy to do in a few years, because cameras that can take pictures of the back of the eye could be run by anyone, could be run by high school students, they're point and shoot. And then, algorithms hopefully will be available, like the artificial intelligence algorithm that I showed for glaucoma detection could autonomously say, you have an 80% risk of having glaucoma. So that's going to be fairly easy to do. Now, we don't have that kind of algorithm yet. It's not yet FDA approved. And there isn't even a CE stamp for such an algorithm in Europe. What we do have are algorithms for detecting diabetic retinopathy, but I think soon we will see such algorithms for glaucoma. So, and I think once that's available, it'll be fairly easy for primary care providers to screen for glaucoma.
Thank you, thank you. Another question, by early onset, do you mean younger people?
Yes, yeah, I think this is part of the other. So I mentioned that one of the dogmas.
You're doing something with your mic and we cannot hear you.
Sorry, can you hear me now?
Yes.
I kind of backed up because I started getting very excited. So just like I said, one of the dogmas that's just not completely true is that glaucoma affects only the peripheral vision first and the central vision later. It's the opposite for a goodly number of patients, and those are the ones that tend to progress. We also tend to think that this is just a disease of old people. And this isn't true. We think that there are a lot of people out there in their 40s and 50s who have glaucoma and don't know it. And for people of color, it's even earlier than that. As I say, our archetype analysis points that the advanced patterns of loss that the computer was able to detect were more common in people of African descent. Those didn't happen overnight. That first patient that I presented with advanced damage was an African American. And so that damage didn't happen overnight. And in my clinical, when I wear my clinical hat, when I see a patient, no matter what their color is, if they have advanced glaucoma and they're fairly young, I say, let me take a look at your children. And I've been shocked at what I found. And we did a study in Little Haiti, south of Miami. We screened for glaucoma among Haitian Americans, and we found that 20% of people between the ages of 20 and 40 had some sign suggestive of glaucoma. That's a lot. So younger people the more we look, the more we're finding open-angle glaucoma in younger populations.
No. Well, then it's a good time to follow up with the next question. And by the way, I'm going to take 2 more questions because I don't want to sort of outstate our welcome here. So the question is, what has been done to analyze mitochondrial development in fetus or newborn that may be related to the decrease later causing glaucoma?
Wow, that's a tough question. I don't know the answer to that one.
Okay.
That's a good question.
Well, we'll come back to it hopefully one day. And last question, I think we've answered pretty much all the questions. Has glaucoma been linked to autoimmune problems? And if so, how can glaucoma be healed?
Yeah, so that's a great question. And the answer is it has been linked to autoimmune problems, both on the goods, it's been tackled from both directions. Mm-hmm. So there was a classic study by Douglas Anderson done in the early 1990s from Bascom Palmer Eye Institute. And he collected all of these patients that had normal tension glaucoma. Pressure, they didn't have elevated pressure, and he was 100% sure that none of these patients had it. And he just asked, compared to controls, were there more autoimmune diseases in the normal tension glaucoma patients? And he found out the answer was yes. We just recently published a paper with my colleagues up at Harvard with Dongfen Chen and Lucy Shen, where they asked the same question, but they didn't limit it to patients with normal pressure glaucoma. They took on all of primary open-angle glaucoma. So you could have normal pressures or you could have high pressures. And they've also found more autoimmune diseases in the cases than in the controls. So there's something going on there. And then there was also studies that suggested that if you were somewhat immunocompromised, if your immune system was somewhat hypoactive, that actually protected you from getting glaucoma. So looking at the Japanese survivors from the bombing at Hiroshima, they found a reduced rate of glaucoma. And when people do animal experiments, if they have animals in a sterile environment or have animals who cannot generate an immune response, and they challenge them by putting microbeads in the eye to elevate the pressure, that is, living in that environment seems to be somewhat protective. So there's something there, Bobby, but exactly how do we leverage that? Because we don't want to give primary open-angle glaucoma patients steroids, which is a non-specific blunter of the immune system, because steroids elevate your eye pressure. So we have to figure out a very, very specific approach, like what exactly it is that's going on. Is it the T cells? Is it the B cells? Is it what's secreted by the cells and could we inhibit that? And people have tried to do that too. So they've looked at TNF blockers. Is tumor necrosis factor is a big potential culprit in mediating glaucomatous damage. And, there's some suggestion that blocking TNF might be helpful for glaucoma, but we haven't been able to leverage that to, Bobby, heal the patients as you've has suggested here. So more work to do to figure out how to leverage that information.
Mm-hmm. So I think it's a good point to stop, unless you can scan the questions and see if there is anything else that we left outstanding.
Yeah, so let's see, there's a question here. Have there been any studies to show that higher prevalence of glaucoma among patients diagnosed with hypertriglyceridemia? The answer
again.
Yep, sorry. The answer is yes. Higher triglyceride levels have been linked to primary open-angle glaucoma. So that's consistent with the findings that I've shown here. So that's one question. And there was another question about, they said that I alluded to lipoproteins, but it would be interesting to look at the lipoproteins in aqueous humor. I totally agree. It hasn't been done. There's been one study that suggests that apoprotein A, which is a component of lipoproteins in aqueous humor, is higher in glaucoma patients than controls. But one needs to do more studies. Studies on aqueous humor are tough because aqueous humor, it's hard to get. Usually you're getting it from patients who are treated, who are at surgery. They have to have their eye anesthetized. They've been treated with glaucoma drops. They've had laser treatment. And so it's hard to sort of figure out the effect of the disease versus the effect of the treatments, but still worth doing. And so more analysis of lipoproteins in aqueous humor is absolutely needed. And I encourage you, Pat, to follow up on that. That's a good avenue of research. Let's see. Those were all the questions I saw in the chat. There's others in here. Am I missing anything?
I think we pretty much answered most of the questions.
Great.
I think we did really well. And I just wanted to thank you, Lou, so much for this fascinating talk, for this look into the future. Hopefully it's going to be bright for glaucoma patients and we'll find many solutions. I would like to thank our wonderful audience who asked questions, who listened to our talk. This talk will be available on our website in a few days. It's recorded and we'll place it on our website. And our constituents, please continue to stay in touch with us. Please continue supporting the Glaucoma Foundation because the research dollars are crucially important to the future of this disease. And on that note, I would like to thank everybody so much again and wish you all a good evening. Bye, everybody.
Bye-bye.